Hello Imbibers, a very good evening to all of you children once are here. Yesterday we discussed about we did a lot of questions related to double fertilization. Today we're going to discuss about the post-fertilization events. Yes, what happens after the egg cell becomes an embryo and of course the respective changes which comes after the fertilization events. Yes, so we're going to discuss all of that today. Before we start, let me introduce you to myself. I am Anam. I'll be your botany expert for the session 2023 and 2024 and we have been discussing everything related to botany, of course the plants, and we'll be discussing in future also. And as you know, we are on the verge of completion of this particular chapter, which is the sexual reproduction in flowering plants. Yes, it is the most important chapter with respect to your NEET examination also, if you ever give it, and your board examination also. So children, all the particular session have been already there on the Embibe dashboard. So please join us on the Embibe app so that you can watch them and you can answer directly through the Embibe app using a laptop, computer, etc. anything you want, okay, which does not give a facility over the YouTube. So children, without wasting any time, let's start our today's session talking about the post-fertilization events. What are the events taking place after once the fertilization has taken place? Yes, next children, the topics to be covered, and we have already covered, we have discussed a lot about these topics already. Yes, we are about on the third last topic. We have discussed about the reproductive parts, we have discussed about the stamen, megasporangium, microsporangium, megasporogenesis, microsporogenesis. We have discussed about the pollination, agents of pollination, we have discussed about the hybridization, we have discussed about the outbreeding devices, pollen-pistil interaction, and what is the unique feature of angiosperms, which is the double fertilization events. We have already talked about it in detail. Yes, and today we're going to see what exactly happens once the fertilization has already taken place, and after that, for the next session, we have scheduled for the seed and fruit formation, and then we're going to discuss about the apomixis and the polyembryony. Yes, so moving forward, let's see what we have to discuss today. That the concept today would be covered would be related to the post-fertilization event, which is the embryo development, dicot embryo, endosperm, embryonal axis, epicotyle, plumule, hypocotyle, radicle, root cap, monocot embryo, scutellum, coleoriza, coleoptile. Okay, all these things, all these small events which takes place after the fertilization will be discussed. So without wasting any time, let's start our today's session. The very first thing, children, talking about a quick recap what we have studied in the previous session. Yes, we have discussed about the pollen-pistil interaction, we have discussed about the fertilization events. We know that in the double fertilization, one male gamete go and fuses with the egg cell and the other male gamete go and fuses with the central cell. Okay, the egg cell after fusion converts into a zygote, which eventually converts into an embryo, while the other male gamete which fuses with the central cell containing two polar nuclei converts into PEN, primary endosperm nucleus, which have a ploidy of 3N. Yes, so this is what we have studied about, and it is called as double fertilization and triple fusion. What is a triple fusion? Because your one male gamete is going to fuse with the central cell which has two nuclei. So three nuclei are present, and that is why it is called as triple fusion. Yes, so this is all we have discussed in the previous session. Yes, and until and unless the pollen is compatible with the stigma does not generate the pollen tube. The pollen is not able to germinate on the stigma if there is no compatibility between them. The rejection is basically caused by the pistil when the compatibility is not met. Yes, and this happens after the compatibility is met between the pistil and of course the stigma and the pollen grain. Yes, moving forward, discussing about today's lecture, children, that a single cell zygote, yes, zygote, which is basically result of the fusion with the egg cell and the male gamete, results into a whole plant body, a big long plants, trees are being formed because of this. So children, a whole root system, shoot system, everything is being formed by the single zygote. Yes, a single zygote give rise to a whole plant body. Yes, of course, and how have you seen that? Let's see what are the parts of this particular zygote existing, which basically creates the different parts of the plant. Yes, talking about the post-fertilization events, children, which includes, very important thing includes the development of the embryo. You know that after the zygote formation, the zygote converts into an embryo, and after the maturation, the ovules which are present in the ovary, yes, the ovules which are present in the ovary, they convert them into a seed, and the whole ovary converts into a fruit. And if you remember, children, angiosperms are the first plants, the first flowering plants which have a seed enclosed inside the ovaries. Yes, the gymnosperms have the naked seeds, yes, while the angiosperms are the first plants which have covered seeds inside the ovary. So after the fertilization takes place, the seeds convert them into the ovule, yes, and the ovary converts into a fruit. This is what is happening after the fertilization. And what happens to the endosperm? There is an endosperm development, and there is also an embryo development. Yes, eventually the whole embryo will also develop, and there will be endosperm will also helps in the development of the seed. Yes, moving forward, let's drag the slider and see what exactly happens. So children, as you know, this what happens, all the accessory parts of the flower, all the accessory parts of the flower will fall off. Yes, they will fall off when they are not necessary, and your whole ovary is converting into a fruit. As you can see, it is being swollen up. Yes, yes, it is converting into a fruit completely, and this is how it happens. This is how your ovary develops into a whole fruit, and inside this particular fruit there would be ovules which will basically causing the formation of the seeds. The ovules inside this particular ovary which got converted into fruit will be converting into seed. So this is how all the accessory floral parts, calyx, corolla will fall off, and the whole female reproductive part will convert into different kind of a formation will take place. Yes, let's see what happens next after this is taking place. We have an endosperm. All of you know that there is an endosperm which is a result of a triple fusion. So what this endosperm is doing inside the seed? Talking about the endosperm, there are, as you can see, children, the primary endosperm cells forms an endosperm filled with reserved food material. So your endosperm, which is a result of a triple fusion, have a reserved food material. Why there is a reserved food material? The reserved food material is for the nourishment of the embryo. Yes, there is a nourishment needed for the embryo. That nourishment comes from the endosperm. There are two types of endosperm. There is a free nuclear endosperm. All of you have consumed the coconut water. The coconut water is nothing but a free nuclear endosperm. Let's have an elaborate definition, which is a primary endosperm nucleus undergoes successive nuclear division. Continuously the nuclear division is taking place, and there is a free nuclear division which basically without the cell wall formation. Now all of you must have consumed coconut water. That coconut water is nothing but a free nuclear division formation of an endosperm. The endosperm, the nuclei has been continuously dividing without the cell wall formation. The other type of endosperm existing is the cellular endosperm. What is the function of the cellular endosperm? Nuclear division which is cell wall formation. Simultaneously is the process of cellular endosperm, and as this is basically the white flesh. Yes, there is a coconut water and there is a white flesh in the coconut which sometimes you also consume. Yes, that what you call as a malai, that white flesh like of coconut, that white flesh in the coconut is nothing but a cellular endosperm. The difference between the free nuclear endosperm and the cellular endosperm is just that the free nuclear endosperm does not have a cell wall formation, while the cellular endosperm does have a cellular wall formation. Yes, that is what you called as cellular endosperm. The best and the biggest example, a very common example, is the white milk, the milk coconut water and the white flesh of the coconut. Yes, sometimes in an embryo, for the nourishment of the embryo, the endosperm could be completely consumed. Yes, in the nourishment of the embryo. But on the other hand also, sometimes the endosperm is persisting. Yes, persisting because this is how it persisting. The coconut water is what? your endosperm which is persisting. It is not completely consumed by the embryo development. Yes, so children, when the endosperm is completely consumed and basically is being used for the nourishment, then example of such kind of seeds are pea, groundnut and beans. All of you have seen peanut, all of you have seen groundnut, all of you have seen beans. In such kind of seeds, the endosperm is completely consumed. But on the other hand, when the endosperm is not completely consumed, basically the endosperm persists. Yes, such kind of seeds are castor seeds and of course the coconut seeds. Yes, this is where the endosperm is persisting. So this is all about the endosperm. The endosperm is necessary for the development of the embryo. It is providing the nourishment to the embryo. It could be free nuclear, it could be cellular. Okay, in a free nuclear, a continuous cellular nucleus division is taking place without the cell wall formation, while in the cellular there is a cell wall formation. Yes, this is about the endosperm. What happens to the endosperm after the fertilization has already taken place. Next children, talking about more, we have a question for you, that free nuclear division in an angiosperm takes place during which event? Yes, during which event the free nuclear division is taking place? Identify the options. Yes, which free nuclear division is taking place in angiosperms? During which of the event? Pollen formation, embryo formation, endosperm formation or the flower formation? Yes, which among them is responsible for the free nuclear division? Just now we have discussed about it. The correct answer would be the endosperm formation. The free nuclear division, yes, it is basically leading to the formation of this coconut water, is a continuous nuclear division without the cell wall formation. Yes, moving forward with the more discussion, next we have the embryogenesis. Yes, what happens, how the embryo develops after the fertilization. So children, I know this is completely basically present in your NCERT also. So quickly learn that the embryogenesis in dicot plant. What is a dicot plant? A dicot plant will have two cotyledons. Two cotyledons. Yes, yes, plumule and radicle. So children, the zygote embryo development is at the micropylar end. All of you know the micropylar end through which the pollen tube enters. Yes, the micropylar end which has the synergids, which has the phylum of apparatus, which has the egg cell. The zygote divides after the formation of certain amount of endosperm. Why certain amount of endosperm? Because the zygote needed, the whole soul needed for the nourishment of the embryo. How does this whole embryo, how this whole nourishment is basically taking place? What are the stages of the embryo development? The very first stage is a pro-embryo stage. Yes, this is a pro-embryo stage, children. This is a globular embryo, as you can say. There is a heart-shaped embryo and finally there is a mature embryo. So these are the few stages of the embryo development. Yes, there is a pro-embryo stage, there is a globular, continuously division is taking place, as you can see. Children, continuously division is basically taking place. This is how, this is what was called heart-shaped embryo. It's completely looking like a heart also. And this is a mature embryo. And as you can see, there are two cotyledons. Formation has already been taken place. One for the root development, the other for the shoot development. So the embryogenesis in the dicot plants takes place when after such stages has been a part of the embryogenesis. The pro-embryo, the globular embryo, the heart-shaped embryo and the mature embryo. Embryo develops at the micropylar end. As you can see also, the embryo is developing at the, this is your micropylar end. This is your micropylar end, this is your chalazial end. And as you can see children, your embryo is being developing at the micropylar end. This is your heart-shaped embryo and this is a complete mature embryo. Yes, this is how the development of the embryo in the dicot plants is taking place, giving rise to the two cotyledons, which is radicle and the plumule. The radicle will give rise to a root development, while the plumule will give rise to a shoot development. Yes, so this is how embryogenesis is taking place in the dicot plant. Next children, we have a structure of the whole dicot embryo. As you can see, this is what happens after the development. This is a mature embryo of the dicot plant. Yes, let's see What are the features of the dicot embryo? As you can see, consists of an embryonal axis and two cotyledons. Yes, there are two cotyledons. Two cotyledons. Epicotyle, the portion of the embryonal axis above the level of the cotyledons. Yes, this is the epicotyle region. Yes, this is the epicotyle region. It is a portion of the embryonal axis above the level of cotyledons. There is a plumule or the stem tips which develops from the epicotyle. There is a hypocotyle, the cylindrical portion above the level of the cotyledons. Yes, the cylindrical portion above the levels of the cotyledons. The radicle, which as a root develops into a hypocotyle, and there is a root cap, the root tip covered with this structure. So these are the basically parts of the dicot embryo. There is an epicotyle region, there is a plumule region, plumule and radicle, which will give rise to a root and the shoot. The radicle will have a root cap also. Yes, the root tip will have root cap, which is basically covered with the which is covering the structure which restrict the root development. Yes, this is how the whole structure of the dicot embryo is basically taking place. Yes, this is what the structure of the dicot embryo is. The epicotyle region, the plumule region, the hypocotyle, radicle and the root cap. This is what the mature embryo of the dicot looks like. Next, children, we have the structure of the monocot embryo. Yes, there is a monocot embryo and there is a dicot embryo also. Yes, as you can see, this is how your monocot embryo looks like. Children, this possesses only one cotyledon and the embryonal axis. The one cotyledon is enough to give rise to a shoot development and the root development. One cotyledon, yes, that is why the monocots are not that advanced like the dicots. Yes, in the grass family, the cotyledons are called scutellum. Please remember, this question comes up very now and then, that in the grass family, what is you call as a cotyledon? In the grasses family, the cotyledons is called scutellum. Then we have a aileron layer. Children, what is the aileron layer? As you can see, the aileron layer, children, this aileron layer is nothing but helping in the nourishment and the development of the embryo. Yes, this layer of cells beneath the seed coat that surrounds the endosperms. Yes, which is also helping in the nourishment. Next, we have a coleorhiza, an undifferentiated sheet enclosing a radicle and the root cap. This is a coleorhiza, which is basically having the undifferentiated seed. The cells are not differentiated, enclosing in a radicle and a root cap. Since you know, in a monocot, only single cotyledon will give rise to a root and the shoot development. Next, we have the epicotyle, the portion of the embryonal axis above the level of attachment of the scutellum. Yes, this is the portion attaching towards the scutellum. Yes. Next, we have coleoptile, a hollow foliar structure that houses the shoot apex and a few leaf primordia. What is a coleoptile? A coleoptile, children, is a hollow foliar structure. Hollow foliar structure will give rise to the shoot system and a few leaf primordia. What is a leaf primordia? Leaf primordia will give rise to the development of the leaves, the branches. That is how the leaf primordia will give rise to a shoot system. Yes. So children, this is how the coleoptile, which is a hollow foliar structure that houses the shoot apex and the few leaf primordia. And these are all the parts of the monocot embryo. The aileron layer, which give nourishment, which is around the endosperm. The coleorhiza, which is undifferentiated sheet enclosing the radicle. Epicotyle, the portion of basically the embryonal axis above the level of attachment of the scutellum, and the coleoptile, which is a leaf, give rise to the leaf primordia. So these are the basic structures of the dicot and the monocot, which eventually will give rise to different parts of the plant. So this is all happening after the post-fertilization. This is how the development will take place to the post-fertilization events. Next, children, we have a question for you. Arrange the steps of embryogenesis. Yes, how basically the embryogenesis is taking place inside the embryo. Yes, quickly. let me show you how exactly basically the places of this. Children, this is your coleoptile. This is your coleoptile. This is your plumule. This is your plumule. This is your radicle. This is your radicle, and this is your coleorhiza. Yes. Yes, and this is your endosperm. So now you will have a better understanding of this. So this is a plumule, will give rise to a shoot system. As you know, this is where it is mentioned S. This is your coleoptile, basically having a hollow foliar structure, the shoot apex and a few leaf primordia. They will give, both them give rise to a shoot development. Yes, and both of them will give rise to a root development. Yes, this is how your monocot seed will basically take place. Yes, now you know basically the places of the coleoptile, coleorhiza, radicle and plumule. This is your endosperm and this is your aileron layer. This is your aileron layer. Yes, now you will remember the parts and they basically function. So your aileron layer is a layer around the coat that surrounds the endosperm. Your coleorhiza is an undifferentiated sheet enclosing a radicle and the root cap. This is your radicle and the root cap, coleorhiza. Next we have the coleoptile, which is a hollow foliar structure which is basically leading to the formation of leaf primordia. Yes, now you will understand, now you will remember the parts of the monocot embryo. Yes, I hope this is clear. This is about the monocot embryo. Yes. This is about the monocot embryo. Next we have a question for you again. Now you can answer that. Arrange them in the following order. Arrange them in the following order. Yes, quickly. Arrange the steps of embryogenesis from simple to complex form. Arrange them . So the correct answer is, children, yes, the pro-embryo formation will take place, then the globular embryo will take place, then there is a heart-shaped embryo will take place and there is a mature embryo will take place. So this is how basically the embryogenesis will take place. Next, children, guys, this is all about today's session. What we have learned in the today's session? Today's session we have learned about the post-fertilization events. Yes, we have seen how zygote is basically developing the embryo and the primary endosperm nucleus, how basically endosperm tissue is being developed. Yes, you know there is a free nuclear division. There are two types of endosperm, free nuclear and of course the cellular one. In the free nuclear, there is a continuous nuclear division without the cell wall formation, and the cellular one, there is a nuclear division with the cellular cell wall formation. Yes, the endosperm is clearly for the formation of endosperm is always precedes the development of the embryo. The developing embryo passes through the different stages such as the pro-embryo, globular, heart-shaped and stages before the maturity. A mature dicotyledons has two cotyledons. Yes, one is the plumule, the other is the radicle. The plumule will give rise to a shoot system, the radicle will give rise to a root system. Embryos of monocot have only one single cotyledons. Yes, coleorhiza, coleoptile, coleoptile which is a part of a plumule, coleorhiza which is a part of a root, and basically they are led to the development of the monocot. I hope you have understood everything regarding the seed development, which is very basic. Yes, restricting to your board, your NCERT, this is all about, this is totally related to that only. Yes, but before you go children, a quick update for the next session. Do you know that seeds remain alive over the 100 of years? Yes, there are some seeds which remain alive, viable for the 100 of years. Yes, they are in existence, they are known for their existence only. And before you go children, you know this is an amazing Embibe app. Let me introduce you to this app. What you have to do, you have to log in through your phone number or your email ID, go to this page, go for the CBSE, select your examination, go your respective 11 CBSE, your language, done, and you will land on this page. And as you click on the home page button, children, you click on the Embibe live classes. Yes, and as you see, this is your upcoming classes children. This is classes for the next 10 days. Yes, let me, oh I have clicked the wrong, let me do it again. Click on the CBSE 12th standard. Yes, selective language, click on this page, go into the home page button, go with the Embibe live classes. Yes, this is how, this is the basically classes basically going on for the maths and of course the biology. Right now they are going on this. These are upcoming classes, Embibe classes for the next 7 days. You can register yourself for these classes. What you have to do, you just have to go register yourself and you can check out the pre-class activities. You can learn through this app various practical concepts. Every concept, topic related to your subjects will be present on this app. You can learn according to your physics, chemistry, mathematics, biology. You can practice, several questions are already there. Embibe experts curated questions are there, your respective examination questions are there. You can go and attempt this and you can test yourself, you can see how much you have achieved. Next children, this is the Embibe Lens app. What you have to do, you just have to scan the text. It will highlight all the keywords and will show you everything in 3D. These are the two amazing apps. So please join us on the Embibe app. Until then, we meet again for the another topic. Take care. Goodbye. Keep imbibing.
Keep imbibing. We believe in you.